Thank you. Good afternoon, everyone. Myself, Amiya Sahoo from Central Inland Fisheries Research Institute in Paris. So this works particularly on all the open water bodies like rivers, wetlands, estuaries, particularly on the ecological aspect. Thank you once again for inviting me, particularly Dr. Vinay, for inviting me for this presentation and integrating this ecological studies in the river system. So if you look at last few presentations, particularly from the France, so you use the fish and also the macroinvertebrate for assessment of river health. So since more than 15 years, we are working on the river health assessment, particularly taking the biotic communities. So I have been experienced since more than 15 years on impact of dams, how the dams has changed or altered the river ecology system. Also you can say these are the indicators of stress or the pollution stress, maybe. So with that background, and since last more than 7 years, I am representing in the River Valley projects as an expert member, particularly on the fisheries and habitat. With this background, let me talk on my presentation on how the different techniques we use, considering the biotic community for the river health assessment, and that we have done in the river Ganga. It's not moving, please. Can you see? Enter. Now let me start. What does a river mean for a biodiversity people like me? So I'm not a hydrologist or a hydraulic background. So to me, this river looks like it starts with the plankton, benthos, fish, the entire life inside. So how to support this life through hydrological, through hydraulic modeling, and all this. So this is an integration. It is the interdisciplinary research. So integrating the entire things. You cannot define a river as such with the only hydrology, hydraulics, or geomorphology. So the life is an integral part of this. So these are the parameters which support this life. So we are there to how to, we talk about the restoration of river, the rejuvenation of the river. For what? For restoration of river for the lives of the aquatic life. So this is what the main concept, why we look the rivers should be like this. So all the organisms should be intact, particularly in the what are the changes it happens if there is a pollution, if there is a different stressors in that. Now look at this, particularly when we talk about the life in the river, starts with the stress from the upstream with the dams or the barrages. It creates the stress. In the middle stretch, we have lot of industries, pollutions. In the middle zone and the estuarine zone or marine zone, you have all the microplastics, all the pollutions in the latter stages. So this case, both the man-made stressors or anthropogens and also the climate change. So who are the main, the vulnerable for that? The entire aquatic systems. If you look at the fish, they are in the higher reaches of the vertebrate systems, starting from the plankton. So they impacted because of the entire stressors, including the pollutions. So look at the freshwater ecosystem. We particularly talk about the rivers or the estuaries or the lakes. So we have the different, say example, the water extraction for the domestic commercial purposes, for the dams or barrages, and over exploitations. And these are all the drivers. So we have to find out what are the drivers and what are the pressures linked with the different. So I'm not going in depth because we have a short period of time. So if you look at particularly this the point source or non-point source of pollution from the industry and the domestic is only for the housing or the sanitation or for the energy. So we have to see what are the drivers and the pressure linked to this. Now, so to these pressures, this is what they says the genesis. So what are the different techniques we use for the ecological pollution assessments? We go for the biochemical process, physiological process, histopathological process, then individual level, population level, and community levels. So we always talk today we discussed that from your presentation, that abundance, richness, that comes under the population, whether the entire population, the fish or say macrobenthos or the planktons, they are being impacted or not, and at the community level. But if being a scientist from researchers, what happens at the molecular level, at the cell level, that become the, if a particular, you have a point source of pollutions and details. But when we talk about the larger perspective, we go for the population or the community levels. So it is just a superficial when you say that what is the impact to the population or the community level. But when you go for this at the biochemical or physiological process, you are talking about the individuals. Now, the recent, this is what we are working at this moment. So this is unpublished data. We are working on the metagenomics aspects. So what are the stress factors, particularly at the gene or the DNA level, and the recent advances in the eDNA, that is the environmental DNA, how based on this environmental DNA, how the river health monitoring and assessment is doing. So starting from the biochemical process, physiological process, individual, population and community, and finally coming to the environmental DNA aspects. So the entire technique is being used for the river health assessment, particularly whether your population is under stress and how to mitigate for that stress parameters. And look at this picture particularly when we talk about the habitat. So we talk about the different hydrological or geomorphological. So this habitat once get disturbed, the entire biotic community get disturbed. So when we talk about the biotic community, it may be fish, may be benthos, may be planktons. So these are the assessment how we work. And we see that particularly we focus on the habitat aspect. Okay, so once you control over the habitat, then automatically biotic community will be you can manage or you can rejuvenate that areas. And say one example of siltation. If because of siltation, how the spawning ground, the nursery grounds be affected. You look at the right side of the pictures, the pollutions, then the water for the migration and feeding, the fish, the plankton, benthic organisms, they get affected because of the low discharge and the pollutions. And also the macrophytes, they get pollutated, and because of the reduced flow, there will be increase in the temperature, and finally the community structure changes. Now, looking at river Ganga, so few minutes we'll be discussing what is the present scenario in the Ganga, particularly from the ecological perspective, because we have a national mission for clean Ganga since more than 7 years. So whatever the data we are presenting, that is based on that study from the last 6 to 7 years, particularly the river Ganga. As Professor Datta was telling, the river Brahmaputra, and it comes after the river Ganga. So the main the river in the India is the river Ganga and its fisheries and how it is linked with the climate change. Look at here the genesis of the Ganga management. So we have this GAP 1 launched in the 1985, as you know, most of you. And the second, when it comes to the mission Clean Ganga in the 2009, and also the Namami Ganga, that is in 2014. I think 7 IITs were there in the program. And if you look at the Namami Ganga program, so also we are a party to that. So that how to revive the river Ganga program in this. And also still it is phase 3 is continuing. And through that project, the main target is to restore, revive the water quality and the aqua life in the river Ganga. So look at, this is the current picture of, if you have 2023, at this moment, river Ganga have 215 species of fish. This is the recent data of our study. 215 number of fish species in river Ganga, starting from the Gangotri to the Ganga Sagar, the entire stretch. That means the entire river Ganga's stream. And of the 215, you see 188, that is the 206 is the indigenous species. Of 215, 206 is the indigenous species and 9 are exotics. So that is creating problem. Now if there is a pollution, there is a reduction of flow, the exotic species, that means from the outside, some species, these are the indicators that population grows if you are in the stress conditions. So this creates a stressor to the native populations. So once there is a stress of pollution, say water reduction, the exotic species goes up. So that is the threat to the river Ganga. And if you look at the entire river stretch, we starting say in the 1822 to 2023. So there is almost in between that there is a number of species, there is a reduction, but slowly this population may be due to the different programs that the government of India is taking up in the river Ganga. So this is what you can say, the fish diversity has slightly improved and coming back to its original status. So that could be one of the interventions what the Ganga Action Plan has taken. So this is what the reality that is going on in the river Ganga. And if you look at the 73% of the fish are in the freshwater. That means from the Gangotri to the Farakka barrage, we have almost 73% at the freshwater species. Then it goes to the estuarine. So we have to protect those fish species. And this is slightly I'm passing through because there is a need. What is the present scenario of the river Ganga so that we can better manage, or that parameter can be well manipulated. If you look at the upper stretch, we have a golden mahasir. Maybe from the upper stretch, those who are staying, so they may be knowing the mahasir species, which are an indicator species of everything. That is the reduction of flow. When we are talking about the environmental flow, we consider that one flux species for the estimation of environmental flow. So in the upper stretch, we consider the mahasir species like we have a trout. In the upper stretch, we have the mahasir. But we consider golden mahasir as the flux species because based on its depth and the velocity. And today we are talking about the trout spawn habitat. Okay. So similarly, we consider as the mahasir and trout both that spawning habitat to be protected in the upper stretch. Coming to the middle stretch of the river Ganga, we have lot of Indian major carps, catfishes, and more number of small indigenous species. So these species provide livelihood to the millions of poor fishermen. So these are the middle stretches of and if you look at the lower stretches, means towards the estuarine zone, so we have more number of fish, 123 fish species. And if you look at this sleep plot indicates the diversity of the fish. So mostly the lower stretch of the river has more diverse. So if you come from the upper stream to middle stretch and the lower stretch, the entire fish diversity is more in the lower stretch of the river. And very interesting, this presentation does not include my hilsa, which is anadromous, like you have salmon. So we are working on the hilsa, which is a migratory species, how the dams has impacted their migrations. So I have not included this presentation, but we are working on this. But this slide indicates that 65% are the food fish. At this moment, the Ganga has this percentage of food fish, ornamental value, sports fish, and others. So this is from the conservational point of view, this is very important to document what is the current scenario and what are the stressors and to block those stressors. And look at the conservation status. We have total 10 near threatened species. As per the IUCN list, we have 2 vulnerable and 1 endangered. So we have to identify which species are the vulnerable, endangered, and threatened. So we have to conserve those species either in the in situ conservation or in the ex situ conservations. So this program is look at the Tehri. The last sentence, you look at the Tehri and Haridwar, they were more confronted with the maximum abundance of threatened species. That means this location, Tehri, is more of a threatened species. That means whether this dam has the impact or the pollution has the impact. So that needs to be assessed in that. Okay. And in this, if you look at, so what are the aqua life? If you look at the aqua life, the climate impact and the pollution, we have talked about the alteration of habitat, then reduction in the diversity of indigenous species. I am focusing that once there is a disbalance in the indigenous species, then it indicates that your ecosystem is always in danger. So the third point is increase in the exotic species. That means outside fish species and the impaired breeding and feeding regimes, alteration in the food chain and food web, carrier of pathogens and carrier of health hazards. And recently it is under the review that we have reported one Morganella species, which is a urinary tract infection, UTI. So that pathogen has been detected in the fish and it is highly pathogenic nature. So the fish, when it is under also that carries the human pathogenic organisms and also the health hazards, example, the heavy metals, pesticides. So you are also consuming. So these are the what happens to the stress when the level is there. So these are the factors that influence on that. So coming to the climate change, we had also a climate change project for a decade, 10 years. But the data I'm presenting just 10 years, one decade before. So if you look at Allahabad, the Prayagraj, so there is a percentage of, if you look at 1980 to 2010, so over the span of almost 30 years, there is increase in 0.9 degrees centigrade in the Allahabad. And what happens? And the rainfall, look at here, the rainfall patterns, pre-monsoon and post-monsoon. In the monsoon rain, there is a reduction in 7%. And in the post-monsoon, there is a 4% increase in the rainfall patterns. So when there is a disbalance in the rainfall pattern and also the temperature, the breeding biology or the spawning process of the fish changes. So the entire population. And if you look at, this is very interesting part in the climate change perspective. Look at few species which are migrating upstream because of climate change. Geographically shift of warm water gangetic fishes like Puntius ticto, then Gullias to the colder stretches, means those who are looking at say present where the Kanpur, so they are slightly moving in the upstream because of the temperature change in the climate change. And also if you look at Mahasir, which was present first time, it was reported in the Karnal where it was 1% and 1.4% of the total population. That means the fish population, the entire population is shifting from the middle stretch to slightly to the upper stretches. So once they are shifting, means their entire habitat parcels, that is in linkage with the climate change, so and habitat alterations. Okay. Now the second most dangerous element is the exotic species. So once your habitat is under stress, we have all these exotic species. These are not native of River Ganga or India. So they are the exotic species like common carp and tilapia. Look at here, it is dominating the common carp, then it comes to tilapia. That means there is no need, it is autobreeder. This means if you release that fish, it will automatically they will increase the population and decrease the indigenous fish population. How? Because of the competition of their feeding. Because once their population is more, so they will compete with the feeding practice with the native population. So they will reduce the native populations. So that is creating now havoc because this is African species, this is tilapia. This is not like our Asian species. So this is now creating havoc in the river Ganga than common carp. Actually, common carp is from the China, but tilapia is from the Africa. So they are creating now in the river entire Ganga stretch, particularly from the Kanpur to the Varanasi. So that stretch, when there is a stress or the pollution, we are getting more number of this type of exotic species. This is an indicator of the stress in the river health. Okay. So now if you look at, I have given that Haridwar to Boxer, particularly the indices, how we have estimated that exotic population index of exotic species. Based on that, you can say that Varanasi and Boxer is highly impaired. So these two stretches, that means that stretch between the Varanasi to Boxer is highly impaired stretches. So then the different measures we have to take care, whether the habitat modification or the more sewage pollution. So we have to find out the point source and we have to take care of this. So this is what the indicators, that fish-based indicators that the ecosystem is under stress conditions. Now another system is the biological integrity of indices. So I am not that at the tax level or the generally phytoplankton or zooplanktons we use for the assessment or monitoring. But generally we take for the fish because it is easy to identify. As you say that the spawning, the spawn, fingerlings, easily we can say where they are moving, and both at the in situ toxicity study we can do in the laboratory, and also the public can relate to the statements about the condition of the fish community, because we have lot of fishermen all along the Ganga or say Brahmaputra. So from there also secondary data can be collected. So which species was getting, like we were discussing from the fisherman perspective, we can go, we can talk, okay, whether there is a change in shift in the fish population or not. So these are the easy for the taking fish as a model for the assessment. Particularly, we consider the biotic integrity of ecological ecosystem, or IBI index. So this is what the indices we use. Now we are using that, as I said, the metagenomics analysis. But before that, we are using the IBI, which is a very good indicator of the technique for assessment of the pollution. Okay, so these are the, I'm not going in depth because these are the steps, different steps we follow for the IBI protocol, water sampling. Because once, as I was telling, that your experimental design is perfect, then you forget about the results. Your design should be perfect, so automatically the result will come. So we plan or design experimentation with very minutely, and this is what the protocol you follow for the IBI. Okay, so we look after the experimental design is perfect. Now these IBI indices has been applied in the river Ganga, and we have taken what are the different stressors, barrage, dams, then sewage pollutions. So different criteria we have taken into these different sites, and we have observed. So these are, if you look at, these are the site number, which are the zonation. So what are the habitat types? So we have correlated with the habitat with that of the stressors. Okay. So example, the say example anthropogenic stressors, say semi-urban or sewage influence. What are the habitat types? What velocity was high, or boulders or travels. So these are the basic inputs required for the IBA calculation. And we, as I said, almost we have taken 11 stations in the river Ganga. You can say this is entire from the Haridwar, Rishikesh to Gangotri Bay of Bengal. So this indicates, if this is not readable, I think this is not clear. So entire trophic level. So if you look at the biological perspective, you have to estimate the trophic yield, tolerance, native species, exotic. So different criteria are there, and these are the different fish species we have to identify. And like this, we have to develop a matrix that how many number of native species, how many number of benthic, column, intolerance. So these are the criteria used for the different sites for the number of species. Then finally, that matrix will give you the result, and I am not reading out this. And finally, if you look at this one, the ecological integrity evaluation of the river revealed that the middle stretch, particular, as I was telling, that from the Boxer till the Boxer, the middle stretch of the river is marginally impaired, and the sites graded accepted are the lower stretches mostly. That means from the IBI indices, we can categorize the entire river system, how it is the stressors, and how to mitigate and rejuvenate. And I was talking, when you are talking about the sediment, okay, sediment dynamics, geomorphology. So these are the organisms, they remain in the sediment, okay. So once you are sediment transportation, this means particularly if you look at the climate change. So these are sessile nature, they do not move like fish. If you have a pollution, fish will be moving from here to there. But these organisms, they cannot move fast. They remain in the sediment. So these are the best indicator, particularly for the carbon emission, for the climate change, and particularly for the sediment transport and the geomorphology studies. Okay. So, but it is easy because when you are taking a long stretch, say example from the river Ganga, Haridwar, say this up to Gangotri, you cannot take all the this number of samples. So, you fish as well as the benthic organisms, we have also taken this organism as a indicator of the river Ganga. If you look at this indicates the recorded water quality of the gastropod class B. We have divided into the different zones, particularly taking this microbenthic organisms. Then another ecosystem is the plankton diversity indicators. So when you talk about the river, so ecology, so we have to take the planktons, you have to take the benthos, macrobenthos, then you take the fish. So entire food chain, food web, you can estimate. You have ample number of data to correlate with your hydrological and hydraulic model, entire things. And if you look at the river Ganga, we have this number of total entire diversity of the 80 genera and 65 in the upper stretch, lower stretch 80, middle stretch 65, and the 45 estuarine zone is 30 genera. So these are the diversity. When we go for the algal pollution index, so similarly as the benthic pollution indices, similarly we go for the algal pollution indices. exotic pollution indices. Well, it is found that the 20 pollution indicator genera of phytoplankton in the present study in our in the entire river system. And this is what the last month my student they have published, particularly what we have done in the from the coal power plant discharge. So, say example, this is the NTPC power thermal plant. It the discharge comes to the river Ganga. We have sampled all the planktons and benthic organisms, how they are behaving with particularly thermal pollutions. And we have seen that this thermal discharge 1.3% time less chlorophyll than that of the normal. Okay, and some species they can tolerate up to 40 degrees centigrades. Imagine the water temperature 40 degrees, not the air temperature. So this algal could be an indicator when in the climate change when we are talking about this. Okay, so the like this one, the entire Ganga the data we have dedicated 2 volumes have come from the science and culture, particularly 88 volume 9 it was dedicated for the fish, and also for the volume 11 for the benthic and the climate change, where we are the editor in that. So these are the few glimpses of how the biodiversity is important for a particular river health management, and particularly for the hydrology and geomorphological studies. And in addition, we study particularly to look at this picture. This is for the hillside like salmon migratory species. We give the tagging, flight tag and feed tags, and we see that whether the fish is migrating, how to restore the species, because massive, it was a parliament questions at the Uma Bharati time, and now the river minister of Jal Shakti. So how to revive this fish? This fish is not only India. This is transbounded. This is also Bangladesh, Myanmar. This is the entire this species is known as the national fish of Bangladesh. Okay, and also state fish of West Bengal. So through tagging, we are seeing that whether it is passing through the Barrackpore barrage, having the fish log or fish dead. So that is what we have seen, that this fish within 2 days, 48 hours, it migrates 235 kilometers. Imagine, 2 days, it migrates 235 kilometers. And this is what the fisherman, well, I took this clip and it was coming in the Times of India, that this is very apt to say that in the 2 hours, 2 days means 48 hours, the fish could migrate. So that means, imagine the power of the velocity negotiation with the river velocity and the habitat and depth, inter-habitat, how it provides. Okay, and also we are providing, as Professor Datta sir was telling, this is the Barrackpore barrage. This is the last end of before the Bangladesh. This is the river Ganga. So just 10 kilometers above Bangladesh border, we have the Barrackpore barrage, and the water discharge diverts to the Calcutta port. We are putting these cages to propagate, conserve these species in this. So like Norway, they go for the salmon. So we are planning to have this type of facility to study the entire breeding, migrations, and the conservation. Particularly, again, we produce and we ranch in the river so that their population will be increased. So with this, I stop my presentation. Thank you so much. Thank you.
Long live India and France friendship.